Okay, picture this. Yellow tape fluttering in the wind, flashlights cutting through the pitch black, and investigators desperately searching for that ultimate clue. A single strand of hair, maybe a microscopic drop of blood. For decades, we've been told that finding this is basically the end of the line for criminals. Deoxyribonucleic acid, DNA. It's the modern justice system's absolute magic bullet. But welcome to this explainer, where we're going to look right past those dramatic TV reveals and dig into the actual science. We're investigating the ultimate forensic tool to answer one chilling question. How accurate is it really? Think about it. What if the ultimate proof is actually wrong? It's a terrifying contradiction, isn't it? I mean, we treat DNA as the absolute gold standard of justice, right? It's an infallible lock and key that guarantees the right person is going to prison. But what if the very science we trust to solve our absolute worst crimes has hidden vulnerabilities? Vulnerabilities that could literally put an innocent person behind bars? Well, let's unravel this mystery together. Section 1. The myth of perfection. So, let's dive in. The sheer scale of the impact DNA has had since the late 20th century is just staggering. It truly, completely revolutionized criminal investigations. It's cracked decades-old cold cases and identified unknown victims when literally all other hope was lost. And crucially, it's been the key to exonerating the innocent. Groups like the Innocence Project have used DNA to free people who spent decades in prison for crimes they absolutely didn't commit. Because of this incredible track record, society built up a myth. The myth that DNA evidence is completely 100% flawless. Section 2. The science of the swab. To really understand why infallibility is a myth, we have to look at how we actually tell two people apart at a genetic level. Now, you and I, we share about 99.9% of our DNA. But forensic analysts, they focus on that tiny remaining fraction. Specifically, they look at what we call short tandem repeats, or STRs. Think of these as specific regions in our genetic blueprint where sequences of base pairs just repeat themselves over and over. And the number of times these sequences repeat varies significantly from person to person. Because of this, unless you have an identical twin, your STR profile is completely unique to you. In the lab, this forensic process breaks down into 5 pretty clinical steps. Step 1 is collection, gathering that tiny bit of blood, saliva, or skin from a chaotic crime scene. Step 2 is extraction, which is pulling the DNA out of that biological material. But often, the sample we find is just incredibly small. That's where step 3 comes in, amplification. We use a technique called PCR, which basically acts like a molecular photocopier. It makes millions of copies of the DNA so we can actually study it. Step 4 is separation and detection, where we read those unique STRs. And finally, step 5 is the real climax of the investigation, comparing that profile against a known suspect or a massive database like CODIS to see if we get a match. But here's the thing, not all DNA is created equal. Investigators generally rely on 2 different types of evidence. Nuclear DNA is the holy grail. It's found right in the nucleus of the cell, it's highly discriminating, and it's what we use in most cases. But let's say a body has been left out in the harsh elements for years. That nuclear DNA, it might be completely destroyed. In those cases, scientists pivot to mitochondrial DNA. This is found outside the nucleus, and while it's much tougher, it's way less specific. It's shared maternally, meaning you, your mother, and your siblings all share the exact same mitochondrial DNA. So it can point investigators in the right direction, but it's rarely a smoking gun on its own. Section 3. Probabilities, not certainties. Now, when an expert witness takes the stand, they don't usually say, "We found an exact match." They give this massive, definitive-sounding number, like 1 in several billion. This is known as the random match probability. But what you really need to take away from this is that this number is not an absolute truth. It's a statistical model. It relies on population genetics databases and assumptions about how frequently certain genetic traits appear in a given population. So yes, it's incredibly strong evidence, but DNA is inherently probabilistic. At the end of the day, it's a game of astronomical odds. And honestly, that leads to a very dangerous psychological trap in the courtroom called the prosecutor's fallacy. This happens when a lawyer or a jury dangerously confuses the rarity of a DNA match with the probability of a suspect's actual guilt. If there's a 1 in a billion chance that a random person has this DNA, juries often hear, "Oh, there is a 1 in a billion chance the defendant is innocent." But actually, that is scientifically and mathematically false. The DNA only proves the suspect's genetic material was present at the scene. It absolutely does not prove they pulled the trigger. Section 4. When DNA goes wrong. Despite its flawless reputation, DNA profiling actually has some severe limitations. First up, environmental degradation. Things like intense heat, moisture, and UV light can destroy a sample, leaving behind only a partial profile that could technically match multiple people. Second, we have complex mixture interpretation. What if a murder weapon was handled by 3 or 4 different people? Untangling that messy genetic web relies heavily on human judgment. Third, simple laboratory errors. Mislabeling tubes or cross-contamination can totally doom an investigation. Just look at the infamous Phantom of Heilbronn case. European police spent years chasing a female serial killer, only to discover the mysterious DNA belonged to an innocent woman working in the factory that manufactured the very cotton swabs the police were using. And finally, cognitive bias. If an analyst knows the detective strongly suspects a certain person, that human expectation can subconsciously influence how they interpret an ambiguous, mixed DNA result.
Section 5. The Phantom of Touch DNA.
So, the most chilling modern complication in forensic science isn't about broken laboratory equipment. It's actually about everyday life. Touch DNA introduces a truly terrifying scenario. We're talking about genetic material transferred simply through everyday skin contact. Modern PCR technology is so incredibly sensitive now that we can generate a profile from just a handful of cells left behind when someone briefly touches a doorknob or a weapon. But because it's that sensitive, it is incredibly prone to contamination and what we call secondary transfer. Think about this. You shake hands with someone at a local coffee shop. They leave the shop, get into a fight, and drop a knife. Your skin cells, transferred from their hand to the knife, are now sitting at a crime scene you never even visited. This raises literally the most critical question in modern forensics. How did the DNA get there? Versus whose DNA is it? We aren't just struggling to identify the source of the DNA anymore. We're struggling with activity-level interpretation. Finding a suspect's DNA on a victim's shirt used to mean case closed. Now, defense attorneys and scientists alike have to ask, was it transferred during a violent struggle, or was it innocently transferred hours earlier on a crowded subway train? This profound ambiguity has led to real, documented wrongful convictions. Section 6. Justice in the balance. Now, the scientific community isn't just standing still on this. They are desperately working to fix these critical flaws. Modern labs are implementing probabilistic genotyping software like STRmix or TrueAllee. These use advanced math to help eliminate that human cognitive bias when untangling those complex DNA mixtures we talked about. Labs are also adopting strict blind verification procedures, where second analyst reviews the data without knowing a single thing about the suspect or the case. Add in hyper-strict contamination controls and enhanced statistical modeling, and we are slowly working toward a system that is far more transparent and much less subjective. Which brings us to our final thought. DNA should be viewed not as absolute truth, but as strong scientific evidence that must be carefully interpreted within context. Look, DNA is undeniably one of the most powerful tools in modern criminology. It has brought justice to the forgotten and freedom to the wrongly accused. But we have to remember, it is not magic. It requires perfect handling, unbiased mathematics, and most importantly, a really clear understanding of its limits. Justice demands nothing less. Because if the ultimate proof isn't completely foolproof, how many innocent people are still waiting for us to realize our mistakes? Comment your thoughts on this video. Also, subscribe to us to see more videos like this in the future. See you in the next video.